Hello, this is Brian Masters, talking today about the regulation of genes that grow spikes on some of a dragon's cells. In order to display the phenotype for spike growth and development in some of their cells, a young dragon must possess the correct combination of genetic activators and lack certain repressor genes. Arrows, such as those following S, K, and E, are activators, and the intersecting horizontal and vertical lines signify inhibitors, such as those following genes P and I. We will go over scenarios in which one or two genes are negative, aka lacking from the network. The wild type is the normal version of the genetic network, and no genes are absent. P is essential for the inhibition of the I gene, which would otherwise inhibit K. K is necessary to activate E, which then activates the phenotype for spikes. S is also useful for activating P and setting the network in motion. Without the P gene, I will not be inhibited, so it will stop K right in its tracks. Without K, E cannot activate, resulting in a spikeless dragon. Without the inhibitor I in the picture, K, which doesn't require upstream activation, can activate E, thus activating spikes. Now with I absent, what is P doing there? It is nothing to inhibit, so it will not function with this system. Without I or E, K is still present. However, it will not be able to perform its function of activating E, therefore this dragon will not have spikes. With neither S nor K, neither inhibitor's function is relevant, as P cannot be activated and there is no K to be inhibited. E will not be activated because there is no K, so there will be no spikes on this dragon. I said earlier that this network determines spike growth on some of a dragon's cells. Even if a dragon is set to produce spikes, there are methods of regulation to ensure that only occurs in specialized cells. DNA methylation happens when a nucleotide gains a methyl or CH3 group on one of its carbons. Methylation on a cytosine preceding a guanine, CpG, near a gene promoter is known to inhibit gene transcription, so CpG methylation is likely the reason why many of the dragon cells do not transcribe the genes responsible for spike growth. For these pictures of transcription, the main takeaway is that the promoter filled with bright green methylated instances of CpG results in a repressed transcription when compared to the promoter with only a few. Here are our results. Only the wild type and I negative mutant managed to keep their spikes. Thank you.